This is Chapter 9 on Microbial Genetics. This is part 1 of 5, so it's a pretty long chapter. So genetics is just the study of heredity, or how organisms pass on genes from one generation to the next. And this part of biology, of science of genetics, explores 4 different things. So we're going to look at the transmission of biological traits from the parent to the offspring. The second thing, expression and variation of those traits, so looking at mutations. The third part, structure and function of the genetic material. And then 4, how this material can also change, and that again goes back to mutations. We're actually going to start with the third part, looking at the structure and function of the genetic material and how it actually replicates. So how we take one piece of DNA and end up with 2 identical pieces. So again, we're going to start with looking at the structure and function of our genetic material. So the levels of the structure and function of the genome, or the genetic material. So a genome is just the total sum of the genetic material in a cell. And we can look at genomes at the organismal level on the left. So we can look at all of the genes that the organism contains. We can look at the cellular level, which includes all of the genetic material in that cell, all the chromosomes in that specific cell. And then on those different chromosomes, we can look at the chromosome level, so we can just pick out one of those chromosomes and look at what genes are on that one chromosome. And then we can also look at the molecular level. And this is looking at, you pick out a specific gene and you actually sequence that gene out, so you see if the sequence is thymine, guanine, adenine, cytosine, or if it has another sequence to it. So you can actually sequence out these genes and figure out what nucleotides are in there and what order they're in. So microbial genomes, on the previous slide we were looking at a eukaryotic cell. If you go back and look at that cell had a nucleus in it. So microbial genomes in eukaryotic cells, so over on the left-hand side we have a eukaryotic cell, and you can see there's a purple nucleus in the middle. So in eukaryotic cells, the genome includes all of the chromosomes in the nucleus. You can see those. But the genome also includes plasmids, which are tiny little circular pieces of DNA that are found in some fungi and protozoan, and that's on the very far left of the eukaryotic cell. And in addition, if you remember when we talked about eukaryotic cells, they have mitochondria and chloroplast. And mitochondria and chloroplast, they also have a little bit of DNA in them. So they have a little circular piece of DNA. So in eukaryotic cells, you have to include all of the chromosomes, the plasmids, and the mitochondrial and chloroplast DNA that are in that cell. If you look to the right, upper right hand, we have a prokaryotic cell. In prokaryotic cell, their genomes include the circular chromosome, and in addition, prokaryotic cells can also have plasmids, so tiny little additional pieces of genetic material. Then on the very bottom right, we have the viruses. And remember, Chapter 6, we looked at viruses, and you can have DNA viruses or RNA viruses. And even though viruses, again, are not living things, they're not classified in living things kingdom, but they still have DNA and RNA, so we can actually look at that they have genomes in them as well. Okay, so on these chromosomes are divided into sections called genes. And these genes, it's a fundamental unit of heredity. So this gene, or the genes we're going to be looking at, one gene codes for different types of proteins. And we'll look at more detail in that. But a gene codes for, or it gives instructions for the cell on what proteins to make. So you can have many, many genes on a chromosome. And genes, there's 3 different types of genes that are included on the chromosome. So you have the genes that code for proteins that I mentioned. These are called structural genes because they actually have instructions in them on how to make a protein and what that protein is going to look like. In addition to the structural genes, there's genes that code for RNA molecules, which again is a type of nucleic acid. And then the third type, we have genes that can control the gene expression, and these are called regulatory genes. These are genes that can turn other genes on and off, so they can give control. Some cells don't want to express certain proteins, so they turn those genes off, and those cells might want to express other types of proteins or genes, so they turn those genes on, and then the cell can make that protein. So the types of genes that are on the chromosome, so all the genes that constitute that genetic makeup, that genome, that's called the organism's genotype. So genotype genes. It's kind of easy to remember, so kind of put those together, genotype genes. When those genes are actually expressed, so expression of the genotype, this is going to create certain proteins, and it's going to create observable or measurable traits, and that's called a phenotype. And examples of phenotype can be like a color, like, red or blue. A phenotype can be measurable, like the height or size of an organism. So anything that you can observe in the organism or you can measure in the organism, that is the phenotype. And the phenotype, I remember it as physical phenotype, so the physical trait that the organism is going to have. And again, the phenotype is related to the genotype. So the genotype tells you what the phenotype is actually going to be. So genomes in organisms, they can vary in size. So viruses, remember, they can have DNA or RNA in them. Viruses can have really small genomes that only have 4 or 5 genes on that chromosome, on that piece of nucleic acid. E. coli, which is a prokaryotic cell, a bacteria cell, they have a single circular chromosome that has about 4,288 genes. So more genes than a virus, and that makes sense. E. coli is a bigger cell, so it has more genes, a little more complex. And then if we look at the human cell, which is a eukaryotic cell, human cells, each one of your cells has 46 chromosomes packed inside the nucleus, and those 46 chromosomes contain 31,000 genes. So human cells have very large genomes compared to the virus up at the top. Okay. So looking at the structure of this genetic material, our genetic material is DNA, and we're going to focus on DNA a little bit more. RNA will come into play later. So DNA, deoxyribonucleic acid, the basic unit of a DNA structure is called a nucleotide. So these are the monomers, those building blocks. Nucleotides are those building blocks that we're going to be putting together. So on a nucleotide, they have 3 parts to them, and this is the little diagram on the left. So we have a phosphate group. That's one part of the nucleotide. DNA has a deoxyribose sugar in the middle, and then to that sugar you have a nitrogen base attached. And there's 4 possible nitrogen bases. You can have adenine, guanine, thymine, or cytosine. So there's 4 possible nitrogen bases that you can stick onto your deoxyribose sugar on your nucleotide. Okay. So these nucleotides, they're going to covalently bond to each other. So we take, in this example, we have 3 nucleotides on the left and 3 nucleotides on the right. Those nucleotides are going to covalently bond together, and the phosphate of one nucleotide binds to the sugar of our second nucleotide. So the first nucleotide on the left, you have a phosphate sticking down the deoxyribose sugar and an adenine nitrogen base attached to it. So that's your first nucleotide. And then your second one, you have the phosphate, deoxyribose sugar, and a cytosine attached to it. And then the third nucleotide has the guanine, and this is on the left side. So that phosphate and the deoxyribose sugar that hook together, they're going to form a sugar phosphate backbone. And these backbones are going to stick out of the molecule. And DNA, it's made up of 2 strands. So we have 1 strand on the left, and then we have a second strand on the right over here, which is the thymine, guanine, cytosine going from top to bottom. These 2 strands in our DNA, they're going to base pair with each other. And there's special rules on how this pairing occurs. So adenine is always going to pair with a thymine, and they hook up together using hydrogen bonds. Hydrogen bonds are pretty weak bonds, but if you have lots of them, they're actually pretty strong, so they can work together to hold the two strands to each other. Okay, so adenine will always pair with thymine on the other side. Cytosine will always pair with guanine. So A and T go together, G and C go together. And then you can see that on the third one, we have a G on the left and a C on the right. So they have to pair up using those base pairing rules. Once your DNA is paired up together, so starting at the very top on the right of the slide, we have our DNA double helix, so you can see the two strands, the two sugar phosphate strands up here. So that DNA double helix forms a DNA. So now we have all this DNA. In eukaryotic cells, we have to condense down the DNA so it actually fits inside of the nucleus. So how the DNA condenses down, you take your DNA strand and you're going to wrap this purple DNA around the pink histones, and those are proteins. So DNA wraps around the histones, so that purple DNA wraps around the pink histones, and that structure forms a nucleosome. So nucleosome is a DNA and a histone together. These nucleosomes condense down, so now I'm on the bottom right-hand picture, where all the pink circles that have the purple wrapped around them are all condensed down together. This condensed nucleosomes, they form a chromatin fiber. So now I'm on the bottom middle of the picture. We have our uncondensed chromatin fiber. This uncondensed chromatin fiber, it will loop. So we have loosely condensed chromosomes, or the chromatin, it's all looping. Those loops are going to supercoil or condense down even further, and then they will wrap around each other, and eventually we get a chromosome. So chromosomes are very condensed DNA and these histone proteins together. This is looking at, on the left-hand side, this is all the molecules, all the nucleosomes put together. We have a double-stranded DNA. And here again, it shows guanine and cytosine pair with each other from the two strands. Adenine and thymine pair together from the two strands. Okay. So one way to represent DNA is this more molecular formula on the left-hand side. We can also represent DNA as kind of a ladder looking, a twisted ladder, which is in the middle, right here. Or DNA can be represented as a space-filling model, which is on the right-hand side. And you can see the twisting, the gray part of the space-filling model, that's the sugar phosphate backbone. And then inside you have the purple and orange and pink and green. Those are your base pairs, your adenine, thymine, cytosine, and guanine. One thing that I want to point out, so if you look in the middle image right here, the blue strand in the middle image, you can see up at the top there's a 5' right here. So this blue strand, it goes from 5' at the top down to 3' at the bottom. If you compare this to the pink strand is actually going the opposite direction, where it starts at 3' at the top, and the 5' is sticking down at the bottom, right here. So I'm not going to go into detail on the 5' and 3', but just to know that the two strands are going in opposite directions of each other. So one goes 5 to 3, the other one on the other side goes 3 to 5. And this will be important when we get to the replication part. So, replication part. DNA replication, if we look at bacteria, DNA replication, bacteria go through a process called binary fission, where one cell up at the top, they have their one circular pink chromosome. That pink chromosome is replicated, so you can see the blue one that's being created up here. Those two chromosomes separate from each other, and then the cells will separate from each other. So that's the process of binary fission, this reproduction process. So the cell has to replicate its DNA first before it can actually split into two cells. So this overall replication process, replication occurs simultaneously on both strands of your double helix. And it's going to create complementary strands. Okay, so we start up here. We have a parental helix up at the top where it has 2 blue strands. Then there's a replication fork. This is where the 2 blue strands are going to separate from each other. So you just break all those hydrogen bonds holding the 2 strands together, and they get separated. And then one of the blue strands, the other side, is newly synthesized, so the purple one. And then the other blue strand, you get another purple strand on the other side. So this whole process is said to be semi-conservative replication, so at the bottom in the bold. And that means that your new DNA strand that's being created, it's half old, so it's half blue, so half of it is from the parental strand. The other side is newly synthesized in the purple. And you can see that on our other new strand over here. So the other one, it's half blue, so one strand is parental, the other half, the purple one, is newly synthesized over here. Okay. So making these exact duplicates of DNA involves a lot of work. It includes over 30 different enzymes that actually do this replication process. We're just going to look at a few of these enzymes. So replication, it starts off at the origin of replication in your chromosome. So over on the picture we have a blue chromosome, and the 2 blue strands represent the 2 sides of the double helix. So you have a starting point, this green box, that's your origin of replication, your starting point. So at your origin of replication you have an enzyme called gyrase. This is going to relieve any supercoiling in your DNA. So you kind of have to unwind our DNA because it's twisted up in that double helix. This is going to relieve that coiling and make it more straight. After the gyrase relieves the coiling, you have another enzyme called helicase. So this is the second one. Helicase, it's going to unwind and unzip your DNA double helix. So helicase is going to separate the 2 complementary strands from each other. So we're separating those 2 blue strands from each other. You then have primase. Primase is it's a primer. So if you think of when you paint something, you have to put a primer on first before you can put the paint on. And it helps the paint to stick. So primase, it puts on a primer, and your primer is in the form of RNA. So you have an RNA primer at the beginning of your replication fork. So we're kind of getting this process started. And then onto your primer, there's DNA polymerase 3 enzyme that attaches. DNA polymerase 3, this is the enzyme that is going to build up the complementary side, so that purple side of your DNA strand that we looked at on the previous slide. And DNA polymerase 3, it's going to add nucleotides in a 5' to 3' direction, so it goes in one direction. Okay. So because your DNA polymerase 3 can only add nucleotides in one direction, one of your DNA strands is going to be called the leading strand. So this strand is synthesized continuously in a 5' to 3' direction. So you put your primer on, the DNA polymerase attaches to that primer, and then the polymerase can just go continuously on this leading strand. The other side, the other DNA strand in your 2-stranded chromosome that we're working on is called the lagging strand. So the lagging strand is synthesized in the 5' to 3', but it's in short segments. You're basically going backwards. So you have to make it in really short segments. The overall direction, we're going 3 to 5', but we're making little short segments that we're going to connect together next. So on the lagging strand, or on both the strands, we're going to have another enzyme, DNA polymerase 1. This is the enzyme that's going to remove your RNA primers. So we want to replace our RNA with DNA so that the whole chromosome is made up of DNA. So DNA polymerase removes the RNA primers and replaces them with little pieces of DNA. And then on our lagging strand, our last enzyme called ligase is going to link those DNA fragments together on our lagging strand so that we have one continuous strand. What was I going to say? Oh yeah, you should probably watch the crash course video to try to understand the DNA replication a little more. I know I went through it very quickly. Basically, you're going to need to know the enzymes and what their function is during that whole process of DNA replication.